Good day, this is Professor Will, CBMD PhD. Today is Friday, May 29, 2020. It is 9:49:56 seconds a.m. Eastern Standard Time, and this is my continuation on Corona series. Let's get started here. This model mimics SARS-CoV-2 infection in humans. Date May 27, 2020. Source Cell Press. Summary. A mouse model of infection with severe acute respiratory syndrome coronavirus 2. SARS-CoV-2 reproduces features observed in human patients, researchers report. A small animal model that reproduces the clinical course and pathology observed in COVID-19 patients is highly needed. Researchers used CRISPR-Cas9 to generate a mouse model that could express HACE2. According to the researchers, their mouse model has several advantages compared with other genetically engineered mice that express HACE2 for modeling SARS-CoV-2 infection. Instead of being randomly inserted, HACE2 is inserted precisely into a specific site on the X chromosome, and it completely replaces the mouse version of the protein. In addition, this is a genetically stable model with few differences among individuals. Moreover, the viral RNA loads in the lung are much higher, and the resulting distribution of HACE2 in various tissues better matches that observed in humans. After being infected with SARS-CoV-2 through the nose, the genetically engineered mice showed evidence of robust viral RNA replication in the lung, trachea, and brain. The presence of viral RNA in the brain was somewhat unexpected, as only a few COVID-19 patients have developed neurological symptoms. SARS-CoV-2 protein, which binds to HACE2 to enter host cells, was also present in the lung tissue and brain cells. Moreover, the researchers identified the major airway cells targeted by SARS-CoV-2 as Clara cells that produce the protein CC10. Our result provides the first line of evidence showing the major target cells of SARS-CoV-2 in the lung. In addition, the mice developed interstitial pneumonia, which affects the tissue and space around the air sacs of the lungs, causing the infiltration of inflammatory cells, the thickening of structure that separates air sacs, and blood vessel damage. Compared with young mice, older mice showed more severe lung damage and increased production of signaling molecules called cytokines. Taken together, these features recapitulate those observed COVID-19 patients. When the researchers administered SARS-CoV-2 into the stomach, 2 to 3 mice showed high levels of viral RNA in the trachea and lung. The S protein was also present in lung tissue, which showed signs of inflammation. According to the researchers, these findings are consistent with the observation that patients with COVID-19 sometimes experience gastrointestinal symptoms such as diarrhea, abdominal pain, and vomiting. But 10 times a dose of SARS-CoV-2 was required to establish infection through the stomach, then through the nose. Future studies using this mouse model may shed light on how SARS-CoV-2 invades the brain and how the virus survives the gastrointestinal environment and invades the respiratory tract. The HACE2 mice described in our manuscript provide a small animal model for understanding unexpected clinical manifestations of SARS-CoV-2 infection in humans. This model will also be valuable for testing vaccines and therapeutics to combat SARS-CoV-2. Be aware of false negatives in diagnostic testing of COVID-19. Date May 26, 2020. Source Johns Hopkins Medicine. Summary. Researchers have found that the chance of a false negative result when the virus is not detected in the person who actually is, or recently has been, infected, is greater than 1 in 5 and, at times, far higher. One of the most commonly used diagnostic tools, particularly during this pandemic, is the reverse transcriptase polymerase chain reaction test RT-PCR, which uses a person's respiratory sample to detect viral particles and determine if the person may have been exposed to a virus. Laboratory professionals across the U.S. and the globe have used RT-PCR to find out if a person has been infected with SARS-CoV-2, the virus that causes COVID-19. These tests have played a critical role in our nation's response to the pandemic. But while they are important, researchers at the Johns Hopkins have found that the chance of a false negative result when the virus is not detected in a person who actually is, or recently has been, infected is greater than 1 in 5 and, at times, far higher. The researchers caution that the predictive value of these tests may not always yield accurate results, and timing of the test seems to matter greatly in the accuracy. The researchers found that the probability of a false negative result decreases from 100% on day 1 of being infected to 67% on day 4. The false negative rate decreased to 20% on day 8, 3 days after a person begins experiencing symptoms. They also found that on the day a person started experiencing actual symptoms of illness, the average false negative rate was 38%. In addition, the false negative rate began to increase again from 21% on day 9 to 66% on day 21. The study, which analyzed 7 previously published studies on PT-PCR performance, adds to evidence that caution should be used in the interpretation of negative test results, particularly for individuals likely to have been exposed or who have symptoms consistent with COVID-19. Dementia gene raises risk of severe COVID-19. Date May 26, 2020. Source University of Exeter. Summary. Having a faulty gene linked to dementia doubles the risk of developing severe COVID-19, according to a large-scale study. Researchers found high risk of severe COVID-19 infection among European ancestry participants who carry 2 faulty copies of the APOE gene termed E4E4. 1 in 36 people of European ancestry have 2 faulty copies of this gene, and this is known to increase risk of Alzheimer's disease up to 14-fold and also increase risk of heart disease. Now the research team has found that carrying these gene mutations doubles the risk of COVID-19, even in people who had not developed these diseases. The team has previously found that people with dementia are 3 times more likely to get severe COVID-19, yet they are not one of the groups advertised to shield or shelter in place on health grounds. Part of the increased risk effect may have been exposure to high-risk prevalence of the virus in care homes. The team found that people with APOE E4E4 genotype were at double the risk of developing severe COVID-19 compared to those with the common E3E3 form of the APOE gene. The team used data from the UK Biobank study, which collects health and genetic data on 500,000 people. The majority of people in the population and in the sample size have not yet been exposed to the virus. In this analysis, 2.36%, N equals 9,022 of participants with European ancestries, N equals 382,188 had the APOE E4E4 faulty gene, but 5.13%, N equals 37 of those who tested positive for COVID-19. N equals 721 had this gene variant, suggesting the risk is doubled compared to E3E3, 410 per 100,000 versus 179 per 100,000. This is an exciting result because we might now be able to pinpoint how this faulty gene causes vulnerability to COVID-19. This could lead to new ideas for treatments. It is important because it shows again that increasing disease risks that appear inevitable with aging might actually be due to specific biological differences, which could help us understand why some people stay active to age 100 and beyond, while others become disabled and die in their 60s. Several studies have now shown that people with dementia are at high risk of developing severe COVID-19. This study suggests that this high risk may not simply be due to the effects of dementia, advancing age or frailty, or exposure to the virus in care homes. The effect could be partially due to this underlying genetic change, which puts them at risk for both COVID-19 and dementia. Good day, this is Professor Will, CBMD PhD on infectious disease and microbiology. Well, hope you enjoyed listening. Until next time, have a great day.